Yes, so thank you very much for inviting me. What I'm going to talk about today is, as it says there, genetic contributions to cognitive and structural brain phenotypes in older age. And in particular, I'm going to focus on something I've worked on for quite a number of years now, which is apolipoprotein E genotype. So straight off the bat, obviously we're all mostly working from home, and there is a reasonable chance with a newborn that there's going to be ambient background tears. So sorry about that. If you do hear crying, and my wife's dealing with it, the baby's not just sitting in the corner. So what I've always been interested in fundamentally is how do we get from genetic variation to brain structure. So there I am holding a copy of my own brain, which was actually scanned in Western General for a research study. And how do we get from genetics to brain structure and function to cognitive decline? And here's a more recent picture of my own brain structure taken at the ICE facility, and that's a 7T scan, which is very cool. And it's been a long-standing interest of mine to the extent that this is almost exactly the same title I gave for a talk about 8 years ago in Edinburgh Psychiatry. So I'm nothing if not consistent. So ApoE, if you don't know, is it's very what's called pleiotropic, and that means that it has a wide range of different functions. So it's a lipid transporter gene. It moves lipids around the body and the brain. And in particular, it's related to amyloid deposition. So amyloid's the kind of the fatty plaques which accumulate, particularly around the hippocampus, but increasingly throughout the whole brain. And it's very common and pathological in Alzheimer's disease. So in the ApoE gene locus, everyone has 2 copies of any of E2, E3, or E4 genotypes. So you can have 2 copies of E2, 2 copies of, you can have a E2, E3, E3, E4, whatever. And 1 copy of E4 increases your risk of Alzheimer's disease by about 3 times, and 2 copies increases it by 15 times. But not only that, it's played quite a kind of important role in our understanding of, or at least the preeminent theory of Alzheimer's disease, known as the amyloid cascade hypothesis, where it's contributed to our understanding that there's this buildup of amyloid, and that starts this kind of cascade of negative things, which leads to neuronal death and shrinkage of various aspects of brain structure, and ultimately dementia. But it's not just the case that increases your risk of Alzheimer's full stop, it's also associated with earlier age of onset of cognitive impairment. Again, where an increased number of E4 alleles is generally negative for risk of cognitive impairment. And amyloid and ApoE are quite a central thing in a lot of big expensive drug trials. So that's what they all try and do. They all try and bust amyloid in the brain, and generally speaking, they don't really work. And that kind of speaks to us not really having a clear enough understanding of exactly what's really going on and at what stage. So there's all these different drug trials and all these different methods to try and leverage our understanding of amyloid and ApoE in Alzheimer's disease, and they generally haven't worked so far. So in previous studies, we've generally shown that ApoE4 genotypes associated with worse cognitive aging in the loading birth cohort, got my mug, with worse brain structure, and also that it's not really offset by lifestyle variant, which it doesn't really interact with lifestyle. So if you have an ApoE4 allele, it doesn't seem to, at least, increase your vulnerability to things like smoking or drinking too much or not exercising. It seems to be just a direct kind of main effect that you can't really offset tremendously by improved lifestyle and so on. So the fundamental questions that interest me, and I'm going to bore you about for the next wee while, are essentially what aspects of brain structure seem to mediate its association with worse cognition, so non-pathological worse cognition like memory, reasoning, and reaction time. Is there evidence of biological differences in people with E4 risk genotype or E2 protective genotype versus the neutral E3? And what evidence is there of gene-by-gene interaction? So I've kind of got a mixture of some relatively older stuff that we've done and also some new stuff, which isn't, one of which is in press and 2 which are completely unpublished and not out yet. So we've kind of run the continuum of the hits versus some new stuff. So starting off with what aspects of brain structure mediate ApoE's association with worse cognition, I'm mostly going to I'm going to focus entirely on UK Biobank from here on out because it's this amazing cohort for what we like to do. So in the past, we've looked at the cognitive test scores in UK Biobank with regard to ApoE genotype, But there's a wee bit of concern about the baseline tests. So these are the baseline tests that the 500,000 UK Biobank participants all completed in between 2006 and 2010. And we've shown in the past that whilst they had to be very novel and quick and computerized, because it was part of a full day of assessments that they did for a range of different mental and physical health areas, and cognitive function was only a small part of that. So the tests were quite quick and novel and so on. So we'd looked at that before, but we were a wee bit concerned about some of the tests are very, very brief. Some of them have a very narrow distribution. There's a degree of floor or ceiling effect where there's one test where very, very few people, well, not very few, but too many people to be appropriate scored no errors, which isn't really what you want. And also the baseline tests weren't really externally validated versus more traditional tests like you might get in the Wechsler adult intelligence scale. So I'm going to go through a paper that we did a couple of years ago now, where we tested for ApoE's association with structural brain health, having said what I've said there about the baseline cognitive tests not being too ideal. So we looked at imaging phenotypes, which we know from previous literature are relatively barn door substrates for worse cognitive abilities. So you could look at the whole wide range. You could do what a geneticist might call a FEWAS of ApoE versus brain structural phenotypes, because there's loads. There's now hundreds of tiny little phenotypes that UK Biobank has derived with regards to brain imaging. But we looked at the heavy hitters. So we looked at white matter tract integrity, hippocampal volumes, white matter hyperintensity volumes, and total grey and white matter. So this is actually shown in the Lothian-Birth cohort. This is the white matter integrity factor. And what this reflects is basically a single score, which to some extent reflects the integrity of the connective white fatty wires which connect different parts of the brain together. And they're a really nice substrate of information processing speed and subsequently overall cognitive ability. The hippocampus, famously shaped like a little seahorse, is associated with a common symptom of Alzheimer's disease, namely declarative memory impairment. And it's an early site of the neuropathological changes that you get in Alzheimer's disease, including amyloid beta accumulations. White matter hyperintensities or lesions are relatively common in aging and reflect demyelination and are a nice marker of cerebrovascular type health. And total grey and white matter, again, nicely shown in Lothian-Birth cohort, is a phenotype which really ages in tandem with cognitive ability as you get older. So if you're not familiar with UK Biobank, it isn't necessarily the case that all of the UK Biobank phenotypes came from the same point. So baseline cognitive data came in 2006, 2010. And then subsequent to that, they have done internet tests, different cognitive tests over the internet, and cognitive tests when people came to the MRI visits around about 2014, 2015 onwards. And that's described in the open access protocol. And also the phenotypes have been described very nicely in a nice paper by Stephen Smith's group, who derived all these phenotypes, and also nicely by Simon. So that's a great paper, and that's looking specifically at the white matter integrity phenotypes. So we looked at, we collated people who had 1 or 2 copies of ApoE4 genotype. So that's E3, E4 versus people who didn't have an ApoE4 allele. We take out people who have E2, E4 because they have a mixture of protective and risk alleles. So we controlled for principal components, which are kind of a measure of stratification. The genetic array that people were tested on, age and sex, deprivation, smoking history, and self-reported cardiometabolic conditions like heart disease, diabetes, hypertension. And we excluded people who self-reported baseline neurological conditions or things like Parkinson's or stroke. And we ended up with a sample of about 9,000 people aged about 62. And this is published about 2 years ago now. We found that there was generally no association between ApoE4 nor an interaction with age on any of those big barn door brain substrates for cognitive abilities. But we did find an association with total white matter hyperintensity. So that suggests ApoE4's effect on brain structure, at least in UK Biobank, seems to be this kind of cerebrovascular route. And again, you can go and find that one a year or 2 ago now. So subsequent to that, we then said, okay, well, we have all these new cognitive tests that were completed at the MRI visit that are much more in line with what people have been using for studies of cognitive ability for a long time. So matrix completion, trail making test, digit symbol. The fluid intelligence test is one of the tests that you could biobank did at baseline, as is a simple reaction time. And then you get tower rearranging, which is kind of similar to like Towers of Hanoi, Towers of London, that kind of thing. So these are much more traditional type cognitive tests. So on the basis of what we knew from that first study in 2017, 2018, and having this new cognitive data, we asked to what extent does ApoE4 associate with worse cognitive ability in the MRI subsample, and does our association with white matter hyperintensities mediate any associations that we find? And generally, so this is reflecting having an ApoE4 allele versus not. There were associations with digit symbol, worse fluid intelligence, and worse tower rearranging. So the tower rearranging score reflects how many people got correct, and these are standardized betas. So you can see that it's really, really small effects. So if you have an ApoE4 allele, you have 0.02 standard deviation worse tower rearranging score in terms of how many you got correct. So it's quite weak. So then we asked, to what extent does the association that we previously showed with white matter hyperintensities, and which incidentally stuck around when we basically doubled the sample size, which is quite cool. And what we found was that it didn't really mediate the associations with digit symbol or fluid intelligence, so kind of executive function type tests, but it did mediate the absolute vast majority of the, well, not the absolute vast, but a significant amount of the association with tower rearranging. So what you have is this relatively nice association and this kind of nice story of ApoE4 seems to associate with worse tower rearranging scores, and a huge amount of that is mediated specifically by white matter hyperintensity volumes. So this comes back to my point earlier on that I quite like instances where we can have a complete story of genetic variation, a particular brain substrate, and a cognitive outcome, so cognitive decline or worse cognitive abilities. But obviously there's a huge amount more to uncover there, and what we could also do is start to look at the much wider range of UK Biobank imaging phenotypes. So moving on to the second thing that interests me, is there evidence of general biological differences in people with E2 or E4 alleles even prior to developing dementia? So there's quite a lot of interest in, obviously, biomarkers for Alzheimer's disease. And ApoE4 is, I think, the best polygenic score for, so polygenic score is a score which accumulates small additive genetic associations with your trait of interest, in this case Alzheimer's disease. The best one to date still only matches the predictive value of ApoE4 in terms of Alzheimer's disease. And there's a huge amount of interest in, are there biomarkers for Alzheimer's disease? I think fairly obviously, and that's relatively self-explanatory. So we went and asked in the whole UK Biobank sample of around about 500,000 people, are there differences in circulating blood biomarkers in people with ApoE4 or ApoE2 genotype who are at theoretically high risk of Alzheimer's disease but haven't developed it yet? Why did we do this? Well, there is conflicting evidence of the effects that different biomarkers can have on the risk of Alzheimer's disease. So in terms of one paper will say something significant, another won't, or the directions will differ. Relatively few biomarkers have been investigated by ApoE genotype status. Most studies had previously focused on one at a time, or relatively few biomarkers in a specific pathway. So no one had looked at just a range of biomarkers en masse. Previous studies were often limited by sample size. So Biobank, famously 500,000 people, is often bigger than previous consortia by some way, and those consortia often have the disadvantage of having collated different phenotypes and different sample characteristics. Whereas with UK Biobank, they are relatively infamously middle-aged, kind of posh, quite bright and healthy. But at least they were all measured in, they had a standard protocol. They were all measured in the same way. And they also have consistent covariates. So in one previous study, they might not control for smoking or deprivation, whereas here we can control for all 500,000 people or thereabouts with the same covariates. It also leads us to the potential to identify more risk factors for Alzheimer's disease, and hopefully things which could inform where treatments should aim. So the UK Biobank biomarker panel came out, I think, 2019, spring 2019. The idea was that they wanted a range of different things you could measure in the blood, which were relevant to a range of different diseases. So not just heart disease, not just dementia, but all sorts. The biomarker panel was led by Naveed Sattar, who's giving a talk online in Edinburgh, well, not in Edinburgh, but you know what I mean. He's giving a talk on Friday, which I strongly recommend. And the idea was really to look at biomarkers which could be relevant to a range of different conditions, and also things which are established risk factors. So low density lipoprotein for vascular diseases, things which are used often as diagnostic measures, so HbA1c for diabetes or rheumatoid factor for arthritis. And also to try and come up with markers which characterize phenotypes which weren't otherwise easy to assess. And you can get all this information on the Biobank website. So here's the document which shows the different range of biomarkers that Biobank provide data on. And again, the link is at the bottom if you want to go and check this out. So we ran ApoE genotype versus each of these biomarkers and adjusted for various what we consider to be important confounders. So the minimally adjusted model accounted for age, sex, baseline assessment centres. So there were 22 centres across the UK which provided UK Biobank data, the vast majority of which is in England. We used 5 principal components for population stratification and again, genotype array. We adjusted for, again, cardiometabolic conditions, and then finally medication, deprivation, and whether or not you'd ever been a smoker. So obviously there's a lot more boring methodological detail in the paper. So these are the radio edit versions of these papers. Again, quite boring but important detail here was there were some biomarkers where a reasonable number of individuals had data not accessible because their values were too low to be picked up by the biomarker kit. And for those individuals, you take their values, assuming that they have data on at least one other biomarker, and then they haven't been recorded as having duff data, as you know, they have usable data. It's just that they had very, very low levels of, in particular, rheumatoid factor, and we recoded them to the square root of the minimum stated detectable value for that assay. So what this figure basically shows is in teal, you can see the ApoE4 effect, and that's basically means higher values for ApoE4. And then the other side is ApoE2, and these are per ApoE4 allele. So what I'm going to do is highlight a couple of very, very basic ones because there's a load of bars here, and you don't need to look at them all. So what this very, very basically shows is in the red, you can see that's apolipoprotein B and low density lipoprotein. So those are some quite large effects, and those are per allele. So what that effectively means is that per allele of E4, there's around about a half standard deviation difference in ApoB, and similarly around about a half standard deviation decrease in LDL, which is, sorry, that way around, no, teal is E4, yeah, fine. So this shows that if you have ApoE2, you have around about a half standard deviation lower ApoB and LDL. What the blue circles, I don't know if you can see my mouse here, but anyway, what the blue shows is that in most cases there was discordance between ApoE2 and ApoE4, which is what you'd expect because ApoE4 increases risk of Alzheimer's disease and ApoE2 lowers it. So what we would roughly expect, in most instances, would be that the directions would be opposing, and that's mostly what we see across all the different biomarkers. Incidentally, this is the finally adjusted model. And in the green, that's cystatin C, and that's a marker of, to some extent, inflammation. It can be a marker of inflammation. So what you really see here is, firstly, that the big association was with lipids, which is kind of what you'd expect with ApoE. And there were some really quite striking effect sizes, particularly with regards LDL and ApoB. As I said, there is discordance between E2 being protective and E4 being deleterious. So in most instances, they go in opposite directions. Supplementary analyses showed that, so these are average per allele effects shown in this figure, but actually when you really looked through the data and really looked at 1 or 2 E4 alleles versus E3, the values did actually show that. And also there was a number of associations with inflammatory biomarkers like C-reactive protein and cystatin C. There are also loads of instances where there were sex interaction between ApoE genotype, sex, and biomarker values. So around about half of the biomarkers showed sex interaction at nominally statistically significant, and those survived correction for false discovery rates, so multiple correction as well. So yeah, there's a number of instances of sex-specific ApoE associations where the effects are bigger in males or females. We did various sensitivity analyses. So the values there show people with dementia taken out. There's about 2,000 people. Well, there's very, very few people who had dementia at baseline. And then about 2,000 people have developed dementia since then, and we chucked them all out. We added the presence of any self-reported inflammatory condition. Something like rheumatoid arthritis, the month of assessment, because there's seasonality throughout the year to some biomarkers, whether or not people were underweight or had obesity. The ApoE association with IGF-1 results were completely unchanged when we controlled for cancer, because cancer can affect IGF-1 levels. And when we absolutely threw the kitchen sink of principal components, which can be a useful control for stratification, it made, again, no difference when we used 40, so all of them, versus just 5. And when we analysed the data in a slightly different way, where initially we chucked out massive outliers, so people who had values 5 standard deviations higher than the mean. Initially we chucked them out, which is very few people. But when you left them in and just did it a different way, the results again were exactly the same. So what does it actually mean? Well, first of all, what was really interesting is that when you looked at the Alzheimer's disease literature, some of the things which were suggested as risk factors for Alzheimer's disease, such as raised IGF-1, which was actually lower here in ApoE4 people, might be due to bias. So some of those studies which report, for example, IGF-1 being raised as a risk factor, those people already had Alzheimer's in that study. And so there might be a degree of reverse causality going on, or bias, which here we've hopefully got around. It reinforces lipid and neuroinflammatory mechanisms in Alzheimer's disease risk. So here we see, in the case of lipid stuff, an absolutely massive association with lipids in between ApoE genotype, and this is in people who haven't developed Alzheimer's yet at all, but they are at substantially increased risk. And it also suggests some biomarkers that with further study and potentially looking sex specific might have some tractability with regards to Alzheimer's disease and cognitive decline one day. So moving on to the third thing which interests me, what evidence is there of gene by gene interaction? So this is really hot off the presses, this one. So about a month ago, there was a really big study which showed that a huge amount of ApoE4's association with Alzheimer's disease can seemingly be offset if you have heterozygosity in the klotho gene, so a 2-SNP diplotype, which can be indexed by one particular SNP. And what they basically showed was that if you're heterozygous for this klotho mutation, you are basically, if you have ApoE4, it's basically half of your risk of Alzheimer's disease goes down. So klotho is quite poetically named. Klotho is one of the 3 fates who spins the thread of human life. She decided when a person was born, thus in effect controlling people's lives. This power enabled her not only to choose who was born, but also to decide when gods and mortals were to be saved or put to death. So a nicely poetic gene. But rather more concretely, it's a transmembrane protein that, in addition to other effects, provides some control over the sensitivity of the organism to insulin. So rather less romantic, that second bit. Anyway, moving on to what they actually found. So they had this, using about 22 independent cohorts and about 20,000 people. What they showed was that the risk of converting to cognitive impairment or Alzheimer's disease, you can see that, I don't know if you can see my mouse pointer, but basically the ApoE at the top here, and then you have no ApoE, so E2 or E3, and then you can see that people that have are heterozygous for this klotho mutation are almost intermediate between the two. And you can index this with one SNP in particular, so RS9536314. So what we did was we said, okay, well, this study has shown that there's a tremendous effect of offsetting ApoE4's negative effects in terms of Alzheimer's disease. In Biobank, we have a tremendous range of cognitive relevant data. We have baseline cognitive data in 500,000 people. We have imaging data. Actually, it's in 40,000 people now. And we have about 2,000 people who have developed Alzheimer's disease or dementia since baseline. So we went in and looked at essentially ApoE versus all of the things that I have shown you previously in this talk. Adjusted for age, sex, assessment centre, principal components, genetic array, deprivation, medication, and smoking history. And essentially what we found was really not a great deal. We found that ApoE associated with risk of Alzheimer's disease, and there were a couple of instances where Klotho genotype associated with Alzheimer's disease, dementia, baseline cognitive ability, brain MRI phenotypes, as I described earlier on the top. But really, we found that there was really no evidence that Klotho specifically associated with any of these phenotypes, or really that there was any interaction whatsoever. So that was disappointing. So a recent study showed that in 20,000 people that this Klotho heterozygosity offset the ApoE4 effect in terms of Alzheimer's disease. We don't really see that supported in the UK Biobank cognitive imaging or dementia data. And this, a common criticism of UK Biobank in terms of neurodegenerative conditions is that they are quite a bit young for it, that they're a bit cognitively healthy, and they're a bit posh and well-educated. Slightly more negative, so that might be that maybe we will start to see more effects as people get older. A slightly more negative and cynical view, however, would be that, so this figure shows ApoE status stratified by TOM40 poly-T repeat status. So this is a poly-T repeat in the TOM40 gene, and it shows really striking results in terms of if you look at, for example, the blue lines. So the blue lines are all people with ApoE E33 genotype, and SVL, SS, VLBL all mean short and very long, and so that's basically how long is your poly-T repeat. And the point I'm making here is that this, about 7 or 8 years ago, was very, very exciting, and everyone thought maybe TOM40 poly-T repeats, this big exciting locus that we could all look at for solving Alzheimer's disease, and it's not really shaken out, pretty much. Also, the fact that we don't really find anything in UK Biobank, to some extent, the Klotho, if you go back to the Klotho figure, theoretically it should really start to kick in around about the age of 80, so 70 to 80, and its effects only become more pronounced. But that doesn't really reconcile with this push. I've got a reference there, but it's from Sperling et al. This idea that there's a continuum of Alzheimer's disease, and it should sort of be evident to some extent in cognitive ability before someone even starts to come up with frank Alzheimer's disease or dementia. So our results don't really correspond to that if Klotho does have a real offsetting effect of ApoE on Alzheimer's disease. So what next? Well, we, having found all those results in terms of the biomarkers, the aim is to look at Mendelian randomization studies, looking at some of these biomarkers versus cognitive ability. We plan to look at also polygenic scores for Alzheimer's disease and some of the biomarkers with regards to brain outcomes like cognitive ability, brain structure, and Alzheimer's disease. And also, we haven't really talked about the fact that there's 20,000 people with repeated cognitive data in UK Biobank. And we just haven't really tended to work on that data very much, but we probably will. So these papers are, the very first one, the 2017 one, we did obviously a couple of years ago, and that's published now. But the other ones, the biomarker paper was led by Amy Ferguson, and that's about to come out in Journal of Alzheimer's Disease. The Klotho one has been led by Rachna Tank, who's a star PhD student, and the ApoE MRI and cognition one was led by Chelsea Dutra, and that's going to be coming out at some point soon too, so keep an eye out for them. A couple of very cynical plugs, because I've hopefully got some of your attention. So the Synapse, what do you call it, ASM is tomorrow, and it's online. So Synapse is this amazing hub for brain imaging researchers, and it's free. You can nip in and out as you find talks interesting, and I strongly recommend it. And also, I'm a member of the Alzheimer's Research UK Demon Network, which is very well named. And it's all about trying to, I hate the word, but synergize expertise in terms of analytics and big data pertaining to Alzheimer's disease. So you should join it because it's full of nice people and there's loads of good content and it's very helpful, I hope, for ECRs. So yeah, thank you very much. Thanks to the Biobank participants and staff, Amy, Ratcher and Chelsea, who did all the real work, and also for inviting me full stop. So cheers. Thanks.
Great, thank you very much for the presentation. Yeah, that was great. Does anyone have any questions? Any questions for Dawn?
Thank you very much for your talk. I feel that I've learned a lot. I was just wondering about the follow-up idea about testing longitudinal intelligence score. As far as I know, I just don't know how to do it practically, because I think a lot of the tests are not very stable. And I guess if you measure longitudinal measures, then it's going to include loads of noise. But then the most stable one, the fluid intelligence, which is actually verbal. Sorry, I can't remember the name.
Verbal numeric reasoning. That's it. Yeah.
Yes, exactly. And I guess theoretically, you would expect it to remain the same, because it's quite supposed to be stable. So I just wonder which measures you would be interested in testing.
Yeah, and another downside as well is that they all got better. They all improved from one test to the next. Yeah, I can't remember off the top of my head, but I would, I think in the PLOS paper, some of the reliabilities were, like the pairs matching one, I think was like, the correlation across tests was like 0.2 or something crazy. So I would probably say, yeah, the verbal numeric reasoning and the reaction time one are probably the best, but yeah, we've not really run much since the repeat data was first made available. So yeah, there's definitely a major limitation to that. I think Tim Salthouse has some stuff about because there's often a reliable increase, as in people get better from wave 1 to wave 2, you can look at how much people improved as a phenotype, I suppose. But yeah, we might look at the repeat stuff, or we might just not. I suppose there's probably repeat cognitive data on some of the MRI tests. So I think about 2,000 people, 1,500 odd, have repeat MRI data. So maybe they've got repeat cognitive test data between those 2 points too. I don't actually know that.
Thank you.
I think the grander point to be made there is that, yeah, a lot of the cognitive data was compromised, and we're very lucky to have it, but some of it's not absolutely ideal for relative to things like the WIMS or the WACE or whatever.
Yeah, I guess the worst is the combination of small sample and then bad tests. At least the sample size is good enough to sort of overcome a little bit of the compromised tests, I guess.
Yes, that's something I found always quite funny, that like, yeah, there are various. So there's like a really good paper by my colleague Breda Cullen, who's generally great, and what she showed was that when you look at things like depression or some mood disorders, a lot of the time people do a bit. The results are just a bit kind of funny. Like, people sometimes do a bit better on the cognitive tests, or effect sizes are very, very wee. And I found it funny that that's in 500,000 people, the cognitive test results can be a bit kind of funny sometimes. But in the 20 odd 40,000 people with the new, really well validated, really sensitive tests, they perform exactly much more the way you'd expect going in. you know, the effect sizes seem to be much more sensitive, and yeah, they just seem to do a lot more what you would expect. So I find that funny, just that it's literally a tenth of the sample size, but you know, having more sensitive tests seems to
matter a lot.
Yeah, yeah.
Thank you so much.
No problem.
Okay, so I guess if there are no more questions, we can end the journal club here.